Welcome. Welcome to my channel Deep Learning Biology. My name is Dr. Vikash Bhardwaj, and in this video we are going to discuss enzyme kinetics. We are going to discuss how to derive Michaelis Menton equation. In this video I am going to teach you how you can derive easily Michaelis Menton equation. It's easy. Be with me and learn how to derive this equation, Michaelis Menton equation. Hopefully you must be knowing what are enzymes. Enzymes, they are involved in carrying out biochemical reactions which are occurring within each of your cell. Now here in the enzyme kinetics we are going to learn rate of enzyme reaction, at which rate enzymes they are going to convert substrate into product. They are going to convert substrate into product. Now here I just want to tell you, keep in mind enzymes generally they are the biocatalyst and they are required in a very minute amount, in nanomolar quantities. But the substrate, it is being required in larger amount. So enzymes, when they are present in nanomolar quantities, they can convert a large amount of substrate into product. Yes, here I also want to tell you that keep in mind enzymes, yes, they can convert substrate into product, but they can also convert product into substrate. So here what is going to happen? Enzyme, it is going to make a complex with the substrate, and this reaction is very much specific. So first initially there is a formation of enzyme substrate complex. So what is going to happen? We have represented the reaction in this way. So first enzyme will form a complex with the substrate, and what is going to happen? There is formation of enzyme substrate complex. So here K1, it represents the rate constant for the forward reaction, means for the enzyme substrate formation. K minus 1, it represents the rate constant for the backward reaction. Yes, the reaction can also proceed in backward. There will be formation of, initially there is formation of enzyme substrate complex, and there might be chance it may break down, enzyme will release, substrate will release without carrying out any reaction. But if the reaction is being carried out in forward direction, what can happen? There can be formation of product also. Enzyme substrate complex, it will break down into enzyme and product will be released. Same way here also you can see the backward reaction. K minus 2, it represents the rate constant for the backward reaction. So this is the overall reaction. Now here keep in mind enzyme, they can carry out both. They can convert substrate into product, they can convert product into substrate. Keep in mind, this is very much important. Now here I just want to tell you, initially in any reaction the concentration of product is negligible. Let's suppose you have started the reaction in which you have added enzyme and substrate. So initially what is going to happen? The concentration of product is negligible. So here what you can do, you can ignore K minus 2, the rate constant for the breakdown of product. You can ignore this. So here keep in mind we are going to ignore this K minus 2. Now our whole equation can be modified in this way. So we have written now this way, we have ignored the value K minus 2. Now here I just want to tell you, we have to calculate at which rate enzyme is working, at which rate enzyme is converting substrate into product. So here the velocity, the rate at which enzyme is working, that can be calculated by K2 into concentration of enzyme substrate complex. So here K2 is the K2 is the rate constant for the breakdown of enzyme substrate complex, and here ES, it represents the concentration of enzyme substrate complex. So here you can calculate the velocity, the rate at which this enzyme is working, by simply multiplying K2, the rate constant, with the concentration of enzyme substrate complex. Now here keep in mind this is equation 1. Keep in mind this is equation 1. Now here you have to calculate the concentration of enzyme substrate complex. Now I just want to tell you, it is not an easy task. It is not an easy task. We have to find the concentration of enzyme substrate complex in a different way. Remember, the concentration of enzyme substrate complex, it will depend on these rate constant K1, K minus 1 and K2. The rate of enzyme substrate complex formation, it will depend on K1, K minus 1 and K2. Now here if you see rate of enzyme substrate complex formation, at which rate enzyme substrate complex will be formed, let's write our equation again. Okay, so here you can calculate rate of ES formation will be K1 multiplied by free enzyme E multiplied by substrate concentration. So rate of ES formation, okay, so here you see enzyme, it is going to bind to the substrate, and K1 represent the rate constant for the ES formation. Okay, so here rate of ES formation will be K1 multiplied by the free enzyme multiplied by the substrate concentration. Now here I just want to tell you, whenever you have added enzyme, okay, so what will happen, it will bind to the substrate, and here let's suppose you have added some quantity of total enzyme. Out of this, some part will bind to the substrate. So you have added total enzyme, out of this some will bind to the substrate, some is going to be free enzyme, and this free enzyme can be calculated easily. Total enzyme total minus the enzyme which have now bind to the substrate, which have formed the enzyme substrate complex. So here rate of ES formation, you can write K1 enzyme total minus E, it is capital S, okay, so please correct this, and the multiplied by concentration of the substrate. So here keep in mind this is equation number 2. This is equation number 2. Now we will calculate the same rate of ES breakdown. So we are going to write this equation again. So here now we calculate rate of enzyme substrate complex breakdown. So it will be K minus 1. Okay, so reaction can happen in backward direction. Okay, K minus 1. Okay, concentration of enzyme substrate complex plus K2 ES. So this way you can calculate rate of ES breakdown. Now keep in mind this is the equation number 3. Now here we are going to assume that there is a steady state. There is a steady state means you have supplied enough substrate, and the enzyme is continuously converting substrate into the product. Okay, so this reaction is happening at a steady state. At steady state, what is going to happen? Rate of ES formation, it will be equal to rate of ES breakdown. So at steady state, what is going to happen? Rate of ES formation rate of enzyme substrate complex formation will be equal to rate of enzyme substrate complex breakdown. So you will now put all the equation. Okay, we have calculated both. Okay, so here you have this rate of ES formation is equal to rate of ES breakdown. Okay, so now we have put both the equation. Now if you solve this, okay, so K1 ET, okay, so you solve this K1 ET multiplied with substrate concentration minus K1 ES substrate concentration is equal to K minus 1, okay, plus K2. You just take out this term common, enzyme substrate complex here. So it will be K minus 1 plus K2 then multiplied by enzyme substrate complex. Now here if you see this term K1 enzyme substrate complex substrate concentration, this term you will take on another side. Okay, so here K1 ET substrate concentration is equal to, okay, it will be, you just take this to the other side, K minus 1 K2 enzyme substrate complex, it will be now in plus K1 enzyme substrate complex and the substrate concentration. Next here you see this term is common, okay, enzyme substrate complex, okay, you take the common term, then solve this K1 ET substrate concentration is equal to K, this you take out this common term enzyme substrate, then here if you solve this K minus 1 plus K2 plus K1 enzyme then substrate, okay, only substrate then in the bracket. So you have just taken out the common term, the enzyme substrate complex. Now here you solve this for enzyme substrate complex, okay, it is equal to K1, okay, ET substrate, okay, then divided by K minus 1 plus K2 plus K1 substrate complex, okay, so this will come out. Next here please write it again, okay, enzyme substrate complex, I am writing this the same term again, okay. Now you divide the upper side and the lower side by K1, okay, so here we have divided both upper side and the lower side by K1, okay, and if you are going to solve this, your enzyme substrate complex concentration will be enzyme total substrate concentration K minus 1 plus K2 divided by K1, and in another K1 will get cancelled, there will be substrate concentration only. Now here in this green color you are going to see K minus 1 plus K2 divided by K1. This is your Michaelis Menton constant known as Km value. So this is your Km value. I will make you understand what is this Km. Plus keep in mind you can represent this Km as K minus 1 plus K2 divided by K1. So here if you solve this enzyme substrate complex, okay, it is equal to enzyme total substrate concentration divided by Km plus substrate concentration. Now here keep in mind, try to recall the equation 1, okay. Now try to recall equation 1, okay, VO is equal to K2 into ES, okay. Now we have to calculate the velocity rate at which this enzyme work, okay. It will be VO is equal to K2 into concentration of enzyme substrate complex. Now if you are going to solve this, you just put the value of ES, concentration of ES here, okay. It is K2 enzyme total substrate concentration Km plus substrate concentration. Now keep in mind this is your equation number 4. Now here if you plot the graph, VO the rate at which enzyme is working with substrate concentration, Remember, initially the rate at which enzyme is working is slow, and as you are increasing the concentration of the substrate, the enzyme will reach its maximum velocity. Okay, so Vmax here represent the maximum velocity of the enzyme at the maximum rate at which the enzyme is working. Now here keep in mind, Vmax is the point where total all the enzyme which is present within the system, it has formed the ES complex. So at Vmax, what is going to happen? Enzyme, the total enzyme is making the ES complex. Now here you can remember, Vmax, it is equal to K2 into ET. What is Vmax? K2 into ET means all the enzyme is now involved in making the ES complex formation. So here we can modify our equation. Okay, so here equation 4, it is now modified. Okay, VO is equal to K2 into ET into substrate concentration. Okay, so this was our equation 4. Okay, now we have seen, okay, K2 into ET. K2 into ET is Vmax. Okay, just you modify this. VO is equal to Vmax multiplied by the substrate concentration divided by Km plus substrate concentration. So this equation, equation number 5, it is Michaelis Menton equation. This is Michaelis Menton equation. Remember, VO, it is going to represent the initial velocity. Vmax is the maximum velocity, maximum rate at which the enzyme is going to work. S is the substrate concentration divided by Km, Michaelis Menton constant, plus substrate concentration. This is Michaelis Menton equation. Now here at the point when Vmax is equal to, VO, the velocity at which enzyme is working, is equal to Vmax by 2. So you know there is a one maximum point. So when VO is equal to Vmax by 2, so you put this in this Michaelis Menton equation. Vmax by 2 is equal to Vmax into substrate concentration, Km plus substrate concentration. Now both Vmax they will get cancelled. Half is equal to substrate concentration Km plus substrate concentration. If you solve this, Km plus substrate concentration is 2 multiplied by substrate concentration. Then if you solve this, Km is equal to substrate concentration. 2 substrate concentration minus 1 substrate concentration, it will remain substrate concentration. So now you can easily understand that the point when VO, the velocity of the enzyme, the rate at which enzyme is working, is half of Vmax by 2. At that point, the Km is going to be equal to the substrate concentration. So here keep in mind, Km, the Michaelis Menton constant, is actually the substrate concentration, at which enzyme is going to have velocity, that is half of Vmax. So here if you are going to plot the graph, so you see that this point is going to represent the maximum velocity of the enzyme. Now here at the point when the Vmax is half of, so here if the VO is equal to half of Vmax, okay, so this point is known as the Km, and Km is going to represent the substrate concentration at which the enzyme has reached its velocity, that is half of Vmax. So hopefully now you have understood what is Km. And keep in mind, VO is equal to Vmax multiplied by substrate concentration divided by Km plus substrate concentration is the Michaelis Menton equation. So hopefully through this video you have learned the basic concept of enzyme kinetics, and in this video you have learned how to derive Michaelis Menton equation. Thank you for watching this. Please do like, share the video. Please do like, share the video and subscribe to my channel. Subscribe to my channel, Deep Learning Biology. Bye bye. Take care.